Friends, today we will learn about biological nitrogen fixation. From today onwards, we will study about plant agricultural biotechnology or plant biotechnology. Different subjects I will teach you through this through different videos. So let's begin with biological nitrogen fixation. The biosynthetic process of reduction of molecular nitrogen to ammonia is called nitrogen fixation. It is a reductive process. Few prokaryotic organisms are capable of nitrogen fixation only. Eukaryotic organisms are unable to fix nitrogen. The biological reaction of nitrogen fixation generates at least one mole of hydrogen, means H2, in addition to 2 moles of NH3 ammonia for each mole of nitrogen molecule. Hence, total 8 electrons are required in reduction of 1 mole of nitrogen to 2 moles of NH3. N2 gives rise to N2 in presence of 8 electrons, reduction of 8 electrons plus addition of 8H plus gives rise to 2NH3 plus H2. However, the actual reduction of N2 occurs in 3 discrete steps. Each involves in an electron pair, N triple bond, N in presence of 2H plus 2 electron minus gives rise to NH, double bond NH gives rise to in the presence of 2NH plus 2 electron minus gives rise to H2N single bond NH2 in presence of again 2 hydrogen plus 2 electron minus gives rise to 2NH3 plus H2. The biological process of nitrogen fixation is catalyzed by an enzyme complex called nitrogenase complex. There are 3 different forms of nitrogenase that differ in their requirement for molybdenum, vanadium, or iron as a critical metallic component. Most of the nitrogenases that have been studied contain a MO factor. Nitrogenase consists of two proteins, a dinitrogenase reductase and dinitrogenase. The dinitrogenase reductase, also called the Fe protein, is a dimer of identical 30 kilodalton subunits based by a 4 Fe minus 4s cluster. Dinitrogenase is a tetramer with two copies of two different subunits. It contains both Fe and MO, iron and molybdenum. Because molybdenum is present in this cluster, the dinitrogenase component is called the molybdenum iron protein, MOFE protein. The MOFE cofactor is the site of nitrogen fixation component, is also called the molybdenum iron protein, MOFE protein. The MOFE cofactor is the site of nitrogen fixation. The genes involved collectively in the synthesis of nitrogenase and the catalytic process of N2 fixation are called NIF gene. Accessory genes are called fixed genes and they are also necessary for the function and regulation of nitrogenase in aerobic nitrogen fixing bacteria. Since nitrogen fixation is a reductive process, so it requires electron donor. In most nitrogenase in most nitrogen fixing microorganisms, reduced ferredoxin acts as a donor for electrons. Ferredoxin is a small 14 to 24 kilodalton protein containing an FeS group. At least 16 molecules of ATP are required for reduction of one molecule of N2. The nitrogenous complex is very sensitive to oxygen. It is irreversibly inactivated by oxygen. Hence, the fixation of N2 must occur under anaerobic conditions. For anaerobes prokaryotic organisms, there is no problem. Facultative prokaryotic organisms such as purple photosynthetic bacteria fix N2 only in anaerobic conditions. In aerobic organisms such as cyanobacteria, anaerobic conditions are created in specialized cells called heterocysts. Heterocysts are thick-walled cells which lack photosystem 2, the oxygen producing photosystem of chloroplast, so they do not generate oxygen. Heterocysts appear to represent an adaptation for nitrogen fixation. Cyanobacteria that lack heterocyst can fix nitrogen only under anaerobic conditions. Symbiotic fixing prokaryotes such as rhizobium maintain a very low concentration of the free oxygen in root nodules of leguminous plants by producing leg hemoglobin, a homologue of hemoglobin. leg Leghemoglobin is present in the cytoplasm of infected nodule cells at high concentrations. Now, let's know about the nitrogen fixing prokaryotes. Nitrogen fixing prokaryotes can be divided into those that carry out biological nitrogen fixation in a symbiotic or a commensalistic relationship with a eukaryote and those that fix nitrogen in a free living state. Most of the nitrogen fixing prokaryotes are free living in the soil. They include photosynthetic as well as non-photosynthetic genera. Only few nitrogen fixing prokaryotes from symbiotic associations with higher plants. Now let's know about the examples of free living nitrogen fixing prokaryotes. Aerobic nitrogen fixing bacteria, that is azospirillum and azotobacter. Anaerobic nitrogen fixing bacteria example is photosynthetic, that is example rhodospirillum. Non-photosynthetic, that is example clostridium. Symbiotic nitrogen fixing prokaryotes may or may not form nodules. The most common form of symbiotic association results in the formation of enlarged and multicellular structures called nodule on the root, the host plant. In case of leguminous plants such as peas, beans, clover, alfalfa, soybean, the symbiote is a bacterium, the host plant. The bacterium of genera Rhizobium, Bradyrhizobium and Azorhizobium. Although most leguminous plants form nodules on their roots, a few leguminous plants bear nodules on their stems. One common example of a stem nodules leguminous plant is Sesbania. Nodules formation also occurs in certain non-leguminous plants such as Ulmus, Myrica and Casuarina. However, the symbiote in this non-leguminous nodule is actinomycetes of genus Franki. Both Rhizobium and Franki live freely in the soil but fix nitrogen only when in symbiotic association with a appropriate host plant. Some symbiotic associations do not form nodules. Anabaina azola, a nitrogen fixing cyanobacterium, lives on the fronds of a water fern called azola. Now, let's know about the genus Phylogenetica. Let's know about some examples of nitrogen fixing bacterial and archaeal genus and their lifestyle. First, I'll tell about genus, then phylogenetic affiliation, then lifestyle. First is Nostoc anabina, that is bacteria. It's free living, aerobic, photolithotropic. Next is Pseudomonas azotobacteria. It's a azotobacter, that is a bacteria, free living, aerobic, chemotrop. Then Thiobacillus, that is bacteria, free living, aerobic, chemotrop. Methanococcus, that is Methanococcus is archaea, free living, anaerobic, chemotrop. Next is chromatium, that is chlorobium, that is bacteria, free living, aerobic, phototrop. Desulfo vibrio, that is clostridium bacteria. This is the bacteria. It's a free living, aerobic, chemotrop. Rhizobium, that is Frankia, it is bacteria, symbiotic, aerobic, chemotrop. Next is Rhizobium. Let's know about the Rhizobium legume symbiosis. Rhizobium is gram-negative, motile, rod-shaped proteobacter that can grow free living in soil or can infect leguminous plants and establish a symbiotic existence. Infection of the roots of a legume with species of Rhizobium genera leads to the formation of root nodules. Chemotactic movement of free-living soil bacteria towards roots is mediated by chemicals such as flavonoids, homoserine secreted by the roots, a specific adhesion protein called ricardacin is present on the surfaces of Rhizobium species secreted by the roots. A specific adhesion protein called ricardacin is present on the surface of Rhizobium species secreted by roots. A specific adhesion protein called ricardacin is present on the surface of Rhizobium species. Ricardacin, a calcium binding protein, play a role in the plant bacterium attachment. Root exudates also induce the modulation, that is node genes responsible for nodule formation. The node genes are classified as common node genes or host specific node genes. The common node genes, node A, node B and node C, are found in all rhizobial strains. The host specific node genes, the common node genes and that is specific node genes such as node P, node Q and node H or node F, node E and node L differ among rhizobial species and determine the host range. Three of the node genes, that is node A, node B and node C, encode enzymes that are required for synthesis of nodulation factors or node factors. Node factors are liposaccharides, the derivatives of chitin. Node factors of all studied rhizobia are composed of a beta-1,4 linked N-acetyl D-glucosamine. The non-reducing terminal sugar moiety is substituted with a fatty acid whose structure varied between different rhizobial species. Node A product of node A of gene node A is a N-acetyl transferase that removes the acetyl group from the terminal non-reducing sugar. Node C product of gene node C is a chitin oligosaccharide synthase that links N-acetyl D-glucosamine monomers. Non-factors induce root hair curling and trigger plant cell division. The process of nodule development begins with the infection of a root hair by Rhizobium bacterium. During the infection process, rhizobia that are attached to the root hairs release node factors that reduce curling of the root hair cells and also sends mitogenic signals and stimulate cell division in cell cortex. Cell divisions lead to formation of root nodule. Let's know about the rhizobia invade the root by digesting the root hair cell wall. After cell wall degradation, the next step is the formation of the infection thread. It is an enfolded tubular extension of the plasma membrane. The infection thread elongates by fusion with vesicles derived from the Golgi apparatus. The infection thread filled with dividing rhizobia elongates through the root hair into the root cortex. Finally, the tip of infection thread fuses with the plasma membrane of the cortical cell, releasing bacterial cells that are packed in a membrane derived from the host cell plasma membrane. In the infected cells, the rhizobia divide and enlarge to form bacteroids, which are separated from the plant cytosol by the peribacteroid membrane. Reduction of nitrogen to ammonia occurs in bacteroid. Nitrogenase enzyme catalyzes this reaction is very sensitive to oxygen. In root nodules, the oxygen level is regulated by leg hemoglobin. It gives legume nodules pink color. The globin protein is encoded by the host plant, whereas the heme group is primarily synthesized by the bacterium and transported into the host cytoplasm. Leg hemoglobin is localized in the cytosol of the infected plant cells and not inside the peribacteroid membrane. Now let's know about assimilation of ammonium ion. NH3, the primary product of biological nitrogen fixation, is toxic to cells in high concentrations. Thus, it is converted into amides or urates. These organic forms are finally transported to the shoot via the xylem. Finally finished. Any questions regarding this, you can ask me in the comment box and subscribe my channel to know more updates. Thank you.